Hi everyone, it's Mrs. Bustamante. Today we're going to talk about 6.1, which is DNA and RNA structure. And really most of this should be kind of a review from our earlier units that we did. So hopefully things sound familiar, but we're going to start to use a lot of it and apply a lot of it. So it's really important that you have a good understanding of it. Here we go. So in this unit, we're really going to talk about how do we get to organisms, kind of, right? We're going to talk about how do we go from DNA to protein and what is it about that DNA that is important, right? So we know that DNA is in chromosomes. In those chromosomes, or rather in that DNA, are genes, and those genes are going to code for proteins. That's eventually where we're kind of going in this unit. So just as we're going through things, kind of keep that in mind about how important it is that DNA is functioning properly and put together properly, and really kind of how powerful it is. So DNA and RNA sometimes in specific organisms like viruses, well, I guess you don't really call a virus an organism, but viruses, RNA is really important in passing along their genetic information, but DNA or RNA is really the source of hereditary information, right? It's going to store that genetic information in those sequences of base pairs in that DNA or RNA, and it's going to pass it on to its daughter cells. Remember, particularly when we're talking about eukaryotic organisms, that DNA is going to get packaged, kind of crunched together, if you will, into chromosomes before they get passed on to their daughter cell. This is all mitosis, meiosis, so that things get the correct amount of information, right? Many viruses actually use RNA in order to encode genetic information and pass that along, if you will. So what makes up DNA and RNA? Well, DNA and RNA are made up of nucleotides. That means they have three parts. They're going to have a nitrogen base. We're going to talk about that nitrogen base, and that's really the part that changes. It can be something A, T, C, G, or U. We'll talk about those nitrogen bases in a second. And it's made up of a sugar, a pentose sugar, a 5-carbon sugar. If we're looking at RNA, it's made up of ribose. The sugar is ribose. If we're looking at DNA, it's made up of deoxyribose. You can kind of think of deoxy as, look, when we compare here hydrogen to an OH, no oxygen, if you want to think about it like that. We're going to go ahead and label these carbons as we go around, and we're always going to start in the same place, right? When we look at our pentose sugar and our nitrogen base and our phosphate, we're always going to say, okay, this kind of looks like a house. We can sort of think about it like that with our oxygen here at the top. This is the point of our house, and we're always going to do one where the nitrogen base will come off. That's always going to be your first carbon. Then you're going to go 2, 3, 4, and 5. That will be how we label the carbons, and that's really important when we go on to figure out how DNA actually replicates. Then they also have a phosphate group that comes off of their 5-carbon in both DNA and RNA. So that was just your quick rundown, right, of nucleotides. I know we did that before, but hopefully that sounds familiar to you. Another thing to remember is that there's base pairing in DNA and RNA, right? DNA is made of polymers, meaning lots of molecules that we put together, monomers put together. And those monomers, if you will, in DNA are nucleotides that we just talked about, right? DNA is going to use adenine, guanine, thymine, and cytosine. And we're going to see that they always pair in the same way. We're always going to see that thymine will pair with adenine in DNA, and that cytosine will pair with guanine. So we say A to T and G to C just because it's shorter, kind of a way to abbreviate them. DNA is therefore going to be double-stranded. And when we get to the part where we synthesize it or make new, we're always going to synthesize in the 5' to the 3' direction. Rather, nucleotides are going to be added to the 3' OH group. Just kind of keep that in mind. You'll notice that we can divide our DNA base pairs into things called pyrimidines and purines. Adenine and guanine are going to be your purines, and your pyrimidines are going to be thymine and cytosine. And you'll notice that purines are always going to be double ringed, and pyrimidines are always going to be single ringed, and that has to do with how we can match them up in a specific pattern. RNA base pairing is just a little bit different. RNA, again, is made of nucleotides, right? But RNA is going to have adenine, guanine, uracil, and cytosine. Notice it doesn't have thymine, so the U kind of replaces the T, if you will. And RNA is also going to be single-stranded. It's still going to be synthesized in the 5' to the 3' direction, meaning nucleotides are added to that 3' OH group. Now, when we look here, uracil is also a single ring, right? So it's a pyrimidine and it's going to replace thymine, so that will structurally kind of make sense to us later on. So this base pairing that we see is what we say is conserved throughout organisms that have DNA and RNA, right? Meaning pretty much all the organisms we look at, right, they contain these 4 base pairs in their DNA. And that makes up a whole lot of differences in the world, right? So I say everyone uses it, right? Because we can see that conserved. We can just see it over and over and over again in all of them. We talked about pyrimidines being single-ringed, right? And here you're single-ringed pyrimidines. And we talked about purines being double-ringed. So nucleotides have a little bit different structure in DNA and RNA, which means that they have how they're put together, right? Which means that they're going to have slightly different functions. When we look at DNA, DNA is going to be double-stranded, and it's going to have adenine and thymine, right? And then when we look at RNA, RNA is single-stranded, and it's going to have uracil, no thymine. So remember that structure for later on. And then quickly, I want to talk about the difference between eukaryotes and prokaryotes. Most of what we just talked about was, you were probably thinking we're talking about eukaryotes, right? We're talking about humans. We spend so much time talking about humans, which are eukaryotes. But remember, prokaryotes also have DNA in them. Now, how it looks is a little bit different. Typically, their chromosomes are circular, which typically means they have slightly smaller genomes. They're not quite as big, right? Because in eukaryotes, we have linear chromosomes, and we know that we pack them up really small, right? And typically, they have larger genomes. Now, both contain these things called plasmids. And these are just small, extra-chromosomal, double-stranded, circular DNA molecules. And we can find them in both prokaryotes and eukaryotes. Where they're found, however, is a little bit different. Prokaryotes, we find them in the cytosol, the liquidy part, kind of think of the cell. And in eukaryotes, we find these plasmids in the nucleus of the cell. So later that's going to kind of become important for us to talk about. But it is also important to know what is the shape of prokaryotic DNA, because I think oftentimes we just go to eukaryotic cells, but remember it's a little bit different, and we can use that and research that too. So that's your basic overview, I guess you could say, or review kind of DNA and RNA structure, and I hope that was helpful.